Today we're going to continue on with our DNA series. In the first two videos in this series, we looked at the discovery of the genetic material, the fact that DNA is in fact what stores and transmits genetic information. There was a big race for that. I encourage you to go back and watch that video. In video 2, we looked at the specific structure of DNA. Now, the structure of DNA really clued in the scientists as to how exactly it was going to carry out its specific functions. How would it store the information? How would it transmit the information? And a very, very big problem that they had prior to the discovery of the structure was they had no way of understanding how in the world does DNA possibly replicate itself. And so in this video, we're going to specifically look at DNA replication. Now, before we kick things off and really get into the nitty-gritty of it, let's talk for a moment. Why must DNA replicate in the first place? Why is it important? Well, here's what happens. In every one of your body's cells, and you have trillions, they all need DNA. Okay, so every cell needs DNA. Because in all of your genome, okay, all the collection of all the DNA molecules within your body, within a cell, there are tons and tons and tons of genes, tons and tons and tons of nucleotide base pairs, these nitrogenous bases that make up the code. And they are needed in order for your cells to function. So these genes may code for the expression of a specific protein that's needed in a specific type of cell. So even though there may be certain genes in your genome that are not necessary for every single cell, it wouldn't be very efficient or very good for your body if only certain portions of DNA were in certain types of cells. Instead, every single cell gets a complete copy of the DNA, and it uses the parts that it needs. And so some genes get turned on, some genes get turned off. And so because every cell needs DNA, we have to replicate it because we can't just split it up. And so there has to be a way that it replicates, okay? And it must replicate before cell division occurs. So every single time any one of your cells divides, it needs a copy of the DNA. Now, when you were conceived in your mother's womb, don't think too hard about that. When you were conceived in your mother's womb, half of your DNA came from your biological father, half of it came from your biological mother. And so when you first came into existence, you are one little itty-bitty tiny cell with a complete set of DNA. Now, that DNA was copied. It was replicated before that very first cell that was you split into two cells. And those cells each copied the DNA before they split into four. And every single time a cell divides, the DNA must be replicated. Okay, so now that we've kind of answered why we need this is very, very important, let's move on and let's take a look at the how. How does DNA replicate? And so I'm going to take, these lovely drawings here are my actually pathetic attempts at interpretation of kind of a double helix of DNA, but I'm going to simplify it down. Notice these two pieces, these little chunk segments of the DNA double helix are tightly wound, they're twisted. Well, when it comes to DNA replication, that twist is actually not always so convenient because, it's very, very, very tightly wound. And so there's actually an enzyme that comes along and helps unwind it a bit and make it a little bit less helical in structure, and that makes it easier to get to the parts that we need. So I'm going to first represent it as a kind of a straight ladder because, again, there is an enzyme that actually helps make it a bit straighter instead of so tightly wound. And so my question to you is this: how will this DNA molecule here, we're going to call this the old DNA, or the original really would be a better word, but I'm just going to write old because it's shorter. We'll say the old DNA, how is it going to be copied into some new DNA. Okay, well, let's take a look here. Here's option number 1. Is this what is going to occur, where we have the old DNA molecule and we literally just completely make a brand new copy of it, just base for base, both strands, we just make everything? Well, that actually turns out to be very problematic for cells. It's not a very good, efficient way to do this, and there's not exactly, the way that DNA is structured, a good way to use the entire old molecule or the original molecule as a template for the new, at least not all at once. So this is a big fat, no, it's not going to work that way. Well, let's look at another option that we have here. What if we copy segments or chunks at a time and break it apart and use that? Well, again, we just don't have a good mechanism by which we can copy these parts. And so one more look here. What if we were to split the original right down the middle into, so that we create two new complete DNA molecules, but they each have an old strand and a new strand, so that part of the original is conserved and the new DNA molecules that we're making have part of the old conserved and a new portion. Well, this is in fact what we see that happens. This is how DNA replication occurs. So we actually begin with the original double helix, we straighten it out a bit, and then we split the hydrogen bonds down the middle between the base pairs, separate it into 2 strands, and then through a rather complicated process, but we're going to try to condense it down in this video. Through a rather complicated process, we add new nucleotides that match, they base pair together with the old strand, complementary. So, without any further ado, let's jump in, and because part of it is conserved, we call this semi-conservative DNA replication, because part of the original strand is conserved when we make the new DNA double helix. So again, we're going to start with this kind of straightened out a bit DNA strand, and you'll notice here, if you'll recall from a previous video when we talked about the structure of DNA, that the strands that make up a DNA double helix run anti-parallel. And what that means is it has to do with the carbons that are part of the deoxyribose sugar in DNA. Over here on this top strand I have drawn here, we have right here the 5' end. That is labeled as 5', meaning that is the location of the 5' carbon in the deoxyribose that would be on the end there. Over here on the other end, we have it labeled 3', meaning the 3' carbon would be on that end of that particular deoxyribose. That would be at that end of the strand. And that is in fact a very important thing, as we'll see here in a moment. When DNA replication occurs, it can only occur from 5' to 3'. The 3' end is always where new nucleotides are going to be added, and that has to do with the way that the enzyme works when it attaches nucleotides. But first, before we even get there, here's what must occur. So we begin with our DNA strand, and the very first step, as you'll notice here, these orange hydrogen bonds here in the middle, I've drawn them in orange, they are going to be broken. Whoop. Hydrogen bonds broken by a special enzyme called DNA helicase. Now, if something in biology, typically if it ends in ASE, it means it's an enzyme. And in this case, helicase looks like, right, helical or helix. It is going to work on the DNA double helix to break apart those hydrogen bonds so that we can access these nitrogenous bases here in the middle. Because what is going to occur is that this strand here, let me switch colors, this strand here and this strand here will both act as templates. These are the strands that will be conserved as we make new DNA double helices. And so to kind of continue on here, each strand, as you can see by the arrows, this strand here is going to head up this way, like I drew there, all right? And it will become the template for a new strand to be added to it by complementary base pairing. The bottom strand will do the same thing. So what you'll notice here is that we are creating the other side of this molecule, going from, you see the bottom here, it's going from 5' towards the 3'. The 3' end right here is always where we add nucleotides. It's always where we add the nucleotides. And so it is working its way this way, attaching new nucleotides using the top up here as its template. The same is occurring down here with the other original strands. It is acting as a template and nucleotides are being added on the 3' end from 5' towards 3'. And so you'll notice that after we split the DNA double helix, replication is occurring, again, anti-parallel. Parallel, but in opposite directions, different orientations. And it is occurring like at the same time, simultaneously, in both directions. So that by the time we get to the end, here we have, again, this is where it was occurring over here on the left. On the right, here is our final product. We end up with two identical DNA molecules. Notice, compare the top strands in each. This one here that goes 5' to 3', and this one that goes 5' to 3'. The only difference you can see in either one, they both go AGA, CTA, AGA, CTA. The only difference you can tell is I colored the backbone white and gray. Gray showing the new strand in that double helix. And so this is known as semi-conservative DNA replication. Again, remember that semi-conservative part of the original DNA strand is conserved and used in the new DNA molecules. Now, this is a very, very simplified version of this process. That's basically it in a nutshell. We unzip it, we take both sides, use them as a template, connect new nucleotides that match by base pairing. A always pairs with T, G always pairs with C. And that's basically it. And you do this along both strands until you reach the end and you have completely replicated that particular chromosome. But of course, like everything in biology, that's a big simplification. There's much more to it. So very quickly, I don't want to make this video too long, but very quickly, let's try to dive into a little bit more of the specifics. So in this diagram, here's what I have. I have in the red, we are representing the DNA double helix here. And actually, again, there's been an enzyme that's kind of straightened it out a bit, unwound it a little bit so that it's easier to work with. And then what you'll see here is there's this big split, this big bubble that has formed within the DNA molecule, and that is the result of these enzymes right here. And again, this is, as I mentioned earlier, DNA helicase. Now I've kind of, you'll see this sometimes represented almost like this triangle wedge kind of shape. It's not really what they look like, but for our purposes here, it makes it a bit easier. And what you'll notice is that these enzymes are splitting, unzipping the DNA molecule in both directions at the same time so that we end up with kind of this big replication bubble here. And the sections where they're actively working right here, where it splits the molecule apart, these are called replication forks. As in like a fork in the road or a fork in the molecule here. And so we're actively in both directions splitting the molecule apart. Well, there's going to be another enzyme that is a major player in this that helps add the new nucleotides. So by the end of this, what we're going to do is we're going to use this top strand here, right? This and this bottom strand here, and we're going to make new DNA. We're going to make new DNA. And so it will be added along here in this direction, and down here it will be added along here in this direction, so that at the end we'll have two completely new strands, two completely new DNA molecules that are, well, I say completely new. They have half of the original, half new, and they are identical, and that's our replicated DNA. But of course, again, it's not just that simple. So let's look. The enzyme that works on this cannot simply just begin adding nucleotides anywhere it wants. It needs some help. It has to be primed and geared up. You know, if you've ever tried to start a weed whacker or weed eater or a lawnmower or something, sometimes some engines, they need to be primed, right? They need some of the fuel mixture pumped into them to kind of get things started. And so kind of the same thing here. We have what is known as RNA primer. And it's literally nothing but a few nucleotides, like 5 to 10 nucleotides of RNA that are complementary. Again, between the red and the yellow is this idea that there are nucleotide base pairs that are complementary and match. And it's literally just to give us a starting point, something to work with, because the enzyme that I'm about to show you needs something to begin working on. Okay, so here's my kind of enzyme. I'm going to show it this way, and imagine it's kind of almost like imagine it's clear. We're looking into it. We can just kind of see what's happening a bit. And so what is going to occur is that this particular molecule, this enzyme, is known as DNA polymerase. Again, ACE is an enzyme, and polymer, this time, polymer is a long chain molecule, so this enzyme helps build the DNA polymer. They name these things really descriptively. It's a good thing. And what it will do is it will begin adding complementary DNA nucleotides that match the nucleotide bases on this red strand, our original strand. But remember, everything occurs from 5' towards 3'. And so it goes in this direction, and it begins adding nucleotides. The blue is going to be my representation, again, of the new part of the DNA double helix. And so it's going to occur in this direction. It has to be added on the 3' end. It has to do with the chemistry of it. That's the only end at which DNA polymerase can attach a new nucleotide. And so it has to occur in that direction. Now, what you're going to notice is, because we have replication forks in both directions, we can actually simultaneously run DNA replication on both sides, both strands. Again, the red is the original. It will be our template for adding the blue, the new DNA nucleotides. They both run in opposite directions, 5' to 3'. But you may be looking at this diagram and thinking, okay, I see it. It's starting here, and it's starting here, and running in this direction. But what about back here? What about this big gap here that's going to form on each side? How will that get filled in? Well, I'm glad you asked. And what occurs with that is this right here. Let me explain this. So where we started and we're running in this direction is called the leading strand. And again, it's going this way. That is the leading strand. It's leading because it's in the front. It's where it started, and it can just keep running this way and kind of keep pace with the DNA helicase at our replication fork. It can just keep going towards the fork. That's the leading strand. It starts out and there's nothing in front of it blocking its path. It can go straight to the replication fork and just continue on with it. But back here behind it, what must occur is that RNA primer must be put down again. There has to be some more RNA primer here. At some point in the past, the replication fork would have been right here with it. Right here where this RNA primer is put down, the replication fork would have been there at some point. But in this case, the replication fork is moving away from the direction, the opposite of the direction of replication. And so it keeps having to add more RNA primer every so often so it can just get going. And again, it runs this direction. 5' towards 3'. And so eventually, this right here, this section right here that starts with a 5' RNA, this will no longer be 5'. It will eventually be gone. But what you end up with are these kind of fragments. It ends up fragmented because of the way that it runs. And so these sections here, see how this blue section is in between the RNA primers? That is known as an Okazaki fragment. It's named for the Japanese scientist who discovered them. And these fragments occur because behind the leading strand, these sections that have to kind of play catch up are called the lagging strands. And so we form these fragments. Now, this actually will occur down here on the bottom as well. Same thing, there has to be some RNA primer put down, and then the DNA polymerase will run this direction, filling that in, and the segment in between, these little segments here that are going to form right here, and there'll be more formed here as it continues on. Those are known as Okazaki fragments, and they will kind of fill in, that's the lagging strand. Eventually, all of that will be taken care of. Eventually, everything will be done. This leading strand will make it all the way to the end of the DNA molecule. Same on the other side. Eventually, the lagging strands will catch up, and these RNA primer sections will be cut out, replaced with the appropriate DNA nucleotides, and it'll be done. And that's it. And the end result is going to be 2 identical DNA molecules, each with an original strand and each with a new strand. And again, these were formed 5' towards 3'. And so they're again, I've just kind of color-coded it here. Old is red, new is blue. But if you were to compare the actual code, the actual bases in this, both of these, this strand and this strand, would be identical. Because we don't want mistakes working their way in there that could lead to things like cancer, cell abnormalities, incorrectly functioning cells, all manner of things. And so your cells actually correct mistakes in the base pairings as it goes. It's very remarkable. And so this is kind of, again, the big kind of summary and a little bit of the details of DNA replication. And so I greatly hope that this video has been a help to you. If you're one of my Scott High School students and you need more help, just reach out to me. And if not, I encourage you to just reach out to me as well. You can comment on the videos. You can message me on my channel. I encourage you to like and subscribe. And as always, thank you for watching Blackboard Biology.